Welcome, everyone, and thank you for joining us. I'm Brenda Dittinger from BioPack, and I will be your moderator on today's webinar, Precision in Research, Synchronizing SuperLab with AcqKnowledge. We will be showcasing some new functionality today. AcqKnowledge and SuperLab now integrate without the need for additional hardware. So we're very excited to showcase this, and we have two great speakers here to give you some demos of SuperLab and AcqKnowledge integration. Today's webinar is being recorded, and we'll send each of you a link once the recording is processed. During the webinar, if you have questions, please submit them through the GoToWebinar control panel. That's how you communicate with me, your moderator. I'll pass questions on to our speakers as much as possible. Before we get to our presenters, I just want to share with you that we host a biennial human physiology training event, and registration is open. In fact, the early bird deadline is just under a month away. That's December 2nd. So if you want that early bird pricing, now is the time to register. The event will be in July 2025 here in Santa Barbara, down the street from our corporate headquarters. We'd love to have you join us. It's a great opportunity to get hands-on experience with all the BioPack devices, including the ones on today's webinar. Here are just a few of my favorite photos from the 2023 event. So save the date, July 14th, and meet us here in California. Now, on to our presenters. We have two wonderful speakers today. Dr. Sanjay Sood has his PhD from the All India Institute of Medical Sciences, New Delhi. He has over 32 years of experience working with a diverse medical student population. As a physiology teacher and researcher, he is currently a consultant with BioPack. Welcome, Sanjay.
Hi, Brenda. Hi, everyone.
We also have Hisham Abood. He founded Cedrus in 1991 to develop software and hardware for the psychology and psychophysiology research community. Their first product was SuperLab, followed in response to increased demand for synchronization of stimulus presentation, EEG, and eye trackers with StimTracker. Welcome, Hisham.
Hello, everyone.
All right, Sanjay, let's get started with you. I'm going to make you presenter.
Thank you, Brenda. Hi, welcome, everyone. So let me share my screen. Here it is. Before we start the webinar, let me give some background information about BioPack. The mission of BioPack is to develop innovative, high quality and competitively priced educational and research tools that allow you to measure physiology anytime, anywhere, on any subject. You see, we have our equipment being used by 99% of the top universities out of 100 top universities in the world, and we are into this business since last around maybe more than 35 years, and our system is being used, and that is quite evident from the citation, research citation we have got, which is more than 53,000 now. And another aspect is that most of the pharmaceutical companies and healthcare technology companies use our products as a gold standard to validate and develop their tools. So having said that, now today's webinar is very interesting. It's really about precision in research. It is the outcome of Cedrus, SuperLab, and AcqKnowledge BioPack, two companies. They have come up with wonderful development for all researchers. Now the SuperLab can communicate with AcqKnowledge over the network. So it is known as NDT, Network Data Transfer. And another very interesting development is that SuperLab and AcqKnowledge can work in the same laptop. So that's also from resources point of view. So you'll be needing only one laptop to work on any of the projects involving stimulus and response. So today the flow is that initially me and Hisham will talk about in general about the equipments we are going to use, and then Hisham is going to demonstrate the building up of SuperLab paradigm. One would be oddball paradigm, another will be Stroop Task. So although we have very limited time, but he'll show you the major steps, and then I'll pick up those same paradigms and try to show that how the stimulus which comes from SuperLab using these paradigm, we can also record various biological parameters. So, and in the end, there'll be question answer session. So just to share with you that MP200 is the BioPack system which was recently launched. It comes with the AMI 100D. If you see, it's attached on the one side. There are around 16 analog channels with two channels which are output channels. So it also has around digital channels and calculation channels, around 8 of them each. It comes with AcqKnowledge software and now also with a cloud-based software, that is AcqKnowledge, and cloud comes in the package. So you can opt for license if you need it. Now, we also have a teaching version, which is a 4 channel, which is known as MP36. So if you see, there are 4 channels, they are universal channels, and it can also give stimulation, uh, like maybe auditory or electric stimulation. So it's a very good tool, very sturdy for teaching, and also some basic research. You can use these tools. And another very interesting aspect is if you have the transducer which you're using for MP36 can also be used by using some gadgets in MP200. Now, BioPacks, as I was telling you, have extensive range of tools for research and teaching. Just to highlight the ones which we are going to use today, we'll be using smart amplifiers. So I'm wired. If you see, I'm wired, and one of the amplifier is EDA, that is electrodermal activity. The other one is PPG. Then I'm also wired. I'm also connected wirelessly, recording my ECG and respiration using bionomadics modules. And then we have also many other gadgets which you can use while recording stimulus response. For example, if you're interested in eye tracking, if you're interested in FNRs, that is cerebral blood flow changes, or any other signals like EMG, EEG. So there are also many um, sensory stimulation options available, from simple haptic, simple touch to scent delivery system. You can go to our website and explore further depending on your research
Right.
requirements.
Hello everyone. My name is Hisham, and greetings from San Pedro, California. We're out beautiful and sunny today. I don't mean to, rub it off in front of anybody, but it's, it's beautiful outside. We're a small company. We've been around for a long time. We founded in 1991, and we specialize in science products. And we do basically just 3 categories of products. And I would like to think we do them well, but yeah. So this is our office. It's an 800 square meters, about 9,000 square feet office. Like a nice quaint neighborhood. From that little office, we've sold products in over 80 countries across the world. It's quite gratifying. And the first category of products that we do is basically response pads. So what you see in here is our Reponda response pads. Um, they're ergonomic. The key tops on them can be changed. The color of them can be changed. It comes with 4 different color caps, and like red, green, blue, etc. And then they also come in 4 different button layouts to suit the different experimental needs. And then the other type of response pads that we do is probably entirely different category, but still a response pad. It's basically MRI compatible response pads. And these are made from 100% plastic and fiber optics. There isn't even a screw in there. The other category of products that we do is products for sending event markers, for example, to the MP200. And so for that, we have different options. A CPOD basically just sends event markers with really high timing precision from your USB to directly to your recorder, whether it's a MP36 or MP160 to 100. And Stim Tracker is a different, you can do the same thing as CPOD, of course, but you can also gather a whole bunch of behavioral data. For example, the onset of a visual stimulus on the screen, or the onset of an auditory stimuli going to speakers, or when a key was pressed on the response pad, etc. And then all these signals are combined, they go to the recorder. The third category, and that's the one that's relevant for us today, is SuperLab, which over time is now a little bit of a product family. SuperLab 6 is very popular in teaching environments, and honestly, one of the most gratifying emails that I get from users is when somebody tells us, I was able to do a 20-minute lecture, and students were building experiments and were getting assignments to make experiments. I really don't think any competing platform can claim that ease of use and ease of this really light learning curve. The next product in our SuperLab family is SuperLab X6, and this is the one that we're going to be talking about today. And that's the one that adds support for BioPack AcqKnowledge, so you can send event markers without needing any hardware. We'll get to that. It can also do gaze contingent presentations with Tobii eye trackers. In other words, it will know live where the gaze is, not after the data has been collected. And then it can also integrate with the LSL, Lab Streaming Layer, which is an increasingly popular protocol out there. And then we also have SuperLab Cloud. This is a COVID baby. So with a SuperLab, with either edition of SuperLab, you can basically click on publish, and then experiment gets uploaded to the cloud. Participants anywhere can run it. They can download a small app, run it. Once they've done running it, the data is automatically uploaded back to your cloud account, and you have all your data in one place. And we are seeing an increasing number of users who are actually using it in lab situations, because it just makes lab management so much easier. You can just get 1 or 2 licenses of SuperLab 6, and you can have, basically as many students as you want run it locally and still have the data come to you. Okay, so that's an overview of the 3 product categories that we do. And now on to today's topic. So there are 2 primary methods of sending event markers to an EEG device or an eye tracker. Okay, so the first one is via TTL signals. 0, 5 volts, really simple stuff. So that's the method that we have been using for a long time. The new method, that's the one that new feature we added to SuperLab X6, is by sending event markers over the network. And there are pros and cons to doing each method, right? So with method 2 over the network, the nice thing about it is SuperLab and AcqKnowledge can run on the same computer, so you have one less computer to deal with. You don't need additional hardware. You also get richer information, and I'll show you that in a second. Why would you still want to use TTL? Well, the main reason to use that is if you are wanting to synchronize data in AcqKnowledge with data on another target device at the same time. Like, for example, Tobii eye trackers. You want both sets of data to be on the same exact time base. You would need to use method 1 for that. But for everything else, really, the BioPack integration with SuperLab is quite nice. So I was mentioning richer information. So this is an example of data. This is actually a screenshot from AcqKnowledge itself. And that shows you the information you're getting. So you have, for example, SuperLab will tell you the name of the experiment, the version that was used when it ran. It will tell you the text that was actually presented. If you're presenting pictures, it will tell you the picture name. You'll know. So that sort of rich information, you cannot really get it with a simple TTL signal. Now, why would we want to do event markers? And I'm sure you all know this, but just so we have a baseline here, you put the cap on a participant's head and you're getting all these signals, analog signals, typically in the microvolt range, really small. And then sometimes it's indistinguishable from noise, but so how do you want to know how to make sense of this data? So by sending event markers, that you see the event markers at the bottom, that from this point on in time, the participant is responding to a stimulus they've seen. So that makes analyzing the data more meaningful by combining the behavioral data with the biometric data. Well, we're going to run 2 experiments, but in the interest of time, we're going to build just one for now. And then Sanjay is going to be running it on his end because he's the guy who has the actual hardware and transducers. So the first experiment we're going to build is an oddball experiment. And in the interest of time, basically, I have prepared the stimuli lists for this one. So a typical oddball experiment, you're presenting 20% targets, 80% non-targets, and you could be presenting, 180 of these things. But over here, we're going to do only a total of 20. So we're going to be doing 4 and 16 non-targets. So I'm going to, when you run SuperLab, this is how the main window in SuperLab appears. It's divided into blocks, trials, and events. And we are going to start by building the experiment. And I'm going to create my first block. And this will simply be instructions. And I need to create a trial also called instructions. And similarly, I'm going to create an event. And this is going to be of type text. And I'm going to just type something in here, say, in this task, you will be hearing 2 different sounds. Press the spacebar when you hear the target sound. Do not respond if you hear the non-target. And that's it. So now I have basically so basically the way it works, we create blocks, trials, and events separately, and then you link them to each other. So now I'm telling SuperLab that this block includes this trial, and then this trial includes this event. And as simple as it is, we basically have an experiment ready to run. It doesn't do anything yet, but it's ready to run. So, okay, we don't want to collect data yet. Okay, that's it. Press a key, this exits. Now, we want to let participants know what a target sound sounds like and what the non-target sound sounds like. So we're going to create a couple of more instructions. Say start target sound, and we're going to let them know you are about to hear the target sound. Press a key when ready. Okay. And then I am going to actually play the target sound. Play target sound. And this is going to be an event of type audio. And I'm going to select a file. Target. Okay. And we want to end this one immediately. We don't want to sit there and wait for a participant response. Something else I want to do over here is I want to erase the message before the sound starts playing. Okay, and then we are going to tell them that they are about to hear the non-target sound, and that's another text message. I copied my text earlier, so I'm just gonna paste it. And now you are about to hear the non-target sound. Same thing in here. I want to erase this message as soon as I press a key. And now we want to play the non-target. Again, it's of type audio. Select a file, non-target. And don't wait for an answer. Or actually, maybe just give them a little bit of time in here. Same thing for this guy. Okay, and then after we do all these instructions, we also need to let them know that the experiment is about to start, so they can get ready. Start experiment. That task is about to start. Press any key when ready. Okay, so I've created these things. So now all I need to do is add them as part of my instructions over here. And if I run this again to get an idea, just to test things out. Now you may not be able, since I'm running on a Mac, the target and non-target sounds, you may not be able to hear them over the GoToWebinar software, but yeah, take my word for them, they're there. But you will hear them when Sanjay runs the experiment on his end. Okay. You are about to hear the target sound, press a key. Okay. Now you are about to hear the non-target sound, press a key. Experiment about to start. All right. So I have my instructions all done, and ball block. Now I'm going to create the real thing. Okay. And the real thing in this case consists of, let me start by creating the event over here. So target sounds. Now, earlier when I selected audio, I was selecting a file, but there's an option in here to tell SuperLab to pick a file from a list. Okay, so this is going to be my target sound, and then I'm going to put for now, I'm going to put a time limit of 1 second for them to respond. Okay, and the correct response for that is the spacebar. Okay, why do we do that? Why do I have to specify the correct response? Actually, you really don't, but if you do, SuperLab in the data file will have a column, and it will be clearly marked C for correct, E for error. So basically, by doing this, you give SuperLab the opportunity to score the responses for you. So it makes it easier to analyze the data later on. So a little bit of preparation in how you do the experiment really helps a lot with the analysis later on. Okay, now let's do the non-target sounds. Non-target sounds. And for this one, I also want to use a list. And my list is the non-targets. And I'm going to put also a time limit for now. And for these fellows, the correct response is to not respond. So we have a go, no go experiment. Okay, so now I need to create a target trials and non-target trials. And I need to tell SuperLab that my target trials consists of this event. My non-target trials consists of this one. And notice how the icon is now looks like a stacked square instead of a regular square. That basically tells you that this is using a stimulus list. And now my block, I want that to include both. Okay, so I have an experiment that's more or less ready to go. It's not exactly 100% done yet, but this is basically the structure of the experiment is done. Really, it's really that simple. However, if I run this experiment right now, all the target trials are going to be presented first, followed by all the non-target trials, and we don't want that. We want to randomize. So to do that, it's basically a couple of checkboxes, and that's it. So now I'm going to get randomized trials. There's one more thing that in designing this experiment that we want to do, which is this guy over here, right? We're giving them 1 second to respond. Subjects, especially when you're running 180 trials, do get habituated. They do know. They will delay their response until the second is almost up. So we want to vary that, to sort of throw them off, make it a little bit less predictable. So we do that in SuperLab using a feature called trial levels. And I'm going to create a one of type time limit. So I'm going to click on that. Let's just call it variable time limit. And in this case, I want to vary it between, let's say, 1 second and 2 seconds. So I'm going to type and that's it. And then level selection by default is randomized. There are many different options on how to handle that. And then when I go back in here, in the input tab, tell SuperLab that this is variable. And then I'm going to click OK next. Go to the next event, the non-target sounds. This too is variable. Okay, so now I have an experiment that's ready to run, and I'm going to run it on my end, but we're not sending event markers yet. So in this task, you'll be hearing two different sounds. You're about to hear the target sound played. You're about to hear the non-target sound. I'm pressing a key. It played. You're about to start. Press a key. Okay. So now it's playing the sounds. I would like to make one small change to this experiment, which is I want to erase this message because if you notice, this stayed on the screen the whole time. So I want to tell SuperLab to just get rid of that. Okay, so my experiment is ready to go. Now, if I want to send event markers to SuperLab, how easy is that? I'm going to show you how. I cannot really keep that turned on because I don't have a AcqKnowledge or BioPack system next to me right now. Sanjay does. So basically, if you want to send event markers, you go to event marking, you say BioPack, you turn that on, and that's it. SuperLab will automatically look for a connection to AcqKnowledge. If I had AcqKnowledge running on the same computer, that almost always says that's successful. It's not an issue. You'll see that on Sanjay's computer. And it's really that simple. Right? So in this case, if I want to run this experiment, I would need to turn that option off because otherwise it's not, it's going to require the presence of AcqKnowledge. And it's really that simple. That's it. Okay. So this is an experiment, a Stroop experiment. You guys are all familiar with that. So in this case, basically, we are presenting words, and the participant is instructed to, if I run it, words and letters will appear on your screen, ignoring the meaning, press the letter. So basically, you just get an answer based purely on the color and not on the word itself or the ink. Press spacebar to proceed. If you notice, there's a cue being presented briefly, and then the word appears. And then the word is being randomized, color is being randomized. Okay. So in this case, this is a little bit different than what we did before. There's here the option, if you want to send event markers via TTL instead of using the BioPack AcqKnowledge, there's an option to add, you can add an event marker. And basically, the way you do that, you create an event just like we did for everything else. You set the event type to digital output. And BioPack equipment supports 8 lines of TTL input. So you select which line you want to activate. And that's typically, for example, line 0 for target, line 1 for non-target, that sort of thing. You can also provide output based on whether the response is correct or incorrect, etc. Then you can also have SuperLab do that for you automatically on certain things. So if you go over here to event marking and you say TTL, you can do things like instead of having to do things for each single event, you can say, okay, on line 6, I want to send an event marker if line is correct. On line 7, I want to send an event marker if line is incorrect. So I'm going to leave that option off for now, since our focus here is the integration with AcqKnowledge, not using TTL markers. But overall, what I'm trying to say is this is another experiment that Sanjay is going to be running on his end, all nicely wired up to to BioPack equipment. Yeah.
Yep.
Back to you.
Okay, so now we'll go ahead and change presenters. Sanjay, I'm going to make you the presenter. And there we go.
Yeah, thank you. Thank you, Brenda. Thank you, Isham, for creating those two experiments, those two paradigms. So what I'll do is I'll use first I'll use my PowerPoint presentation for showing Stroop Task. So before we go any further, let me explain what Stroop means. It means there is interference between color processing and the word processing. For example, when you show a neutral word, it hardly interferes. You just name the ink of the word, that is black, and then in this case red. Now what happens is it becomes a little complex when there is a word and also ink of the word. So you have to identify the ink. So then it becomes little difficult, but if it's congruent, both are same, then it's very easy, fast. But in this scenario, the word is red and the color is blue, so it creates little bit of interference, and that creates little window to understand higher cognitive processes in the lab. So first what I'll do is I'll pick up the cognitive this Stroop task to for live demonstration. So let me show first that I'm wired for today's session. So here if you see, we have MP system with me, MP 160, and sorry, the image is reversed. So Brenda, can we change? Is it possible from your end? No, but that's fine. Now, if you see, this is MP 160 system, and this is bionomedics module. So I'm wired with ECG and respiratory signals. And then we have AMI 100D. So here I'll pick up EDA and PPG and also response switch. So here is the response switch which I'm going to use. And also I have bionomedics module here and respiratory belt. So both are in place. So now the next step would be, let's see the AcqKnowledge component of the setting. Now, as you're aware, BioPack has a AcqKnowledge software which is very versatile. I'll open the window here. So here, if you see, there are around 4 or 5 channels. The first channel will record EDA response, that is the electrodermal activity. So whenever any sudden stimulus comes, there is increased sympathetic output, and that is picked up by these EDA transducers. And then the second one is the respiratory belt, which will pick up inspiration and expiration. I'm also wired, if you see my hands, so I have a PPG transducer here, and these two are for EDA response. And then I also have a hand switch, which is really required for any response activity. When there is stimulus is given, if you have to respond, so what will happen is one of the channels is specially dedicated for the response input. And of course, in the end, you see there is also ECG being picked up. Now, before I go to the actual demonstration with the SuperLab, let me show what is being recorded now. So I'll switch it on. My bionomedics module is on. Let me see. Yeah, it's blinking. And let's see now. So here, the first channel is EDA response, and the second one is respiration. If you see here, when I'm breathing in, so you can also add, you can change the sensitivity. Let me change it a little bit, make it more sensitive. So whenever I breathe in, there is inspiration. So that results in increased sympathetic output, and there will be increase in EDA response. So just focus on the first two channels. So what I'll do is I'll breathe in suddenly. See, there is a shift in the EDA response. Let me change the sensitivity a little more. It's adaptive, so it should show later maybe. Now again, I'll breathe in. Let me stop this here and open again because the sensitivity has changed. So I'll open again and start recording. Okay. So if you see, there is a global panel which is picking up the event signals. So I'm pressing escape. So whenever taking deep inspiration, there is a marker appearing, so which will correspond to the onset of stimulus. And then similarly, if you see in the third channel, I'm picking up my peripheral pulse rate. So it's showing that. And the last one is, second last one is hand switch. When I'm pressing the button, respond to any stimuli like targets, it will be acknowledged here. And the last one is ECG, which is being recorded. So what you can do is you can extrapolate it into maybe heart rate variability analysis and other aspects which can be done. Now let's stop here. Now I'll pick up the SuperLab and the paradigm we created. Technically, if you see, when you do typical uh, Stroop task, there are many steps, many blocks. First, you have to demonstrate to the subject, he needs to practice. Then you give neutral stimuli, and then you give color and words true. But because of the limitation of time, what we have done is we have created a Stroop task with only one block, and that block is color names and description of the ink of the words. So this is what we are going to run today. Now let's see, as Hisham was talking, that connecting SuperLab to AcqKnowledge BioPack is very easy. Yeah, it's true. What you do is you go to the menu, drop down menu experiment, and then go to event marking. So you'll have three options. One is TTL. TTL can be, when you're using response pads, so you can use CPOD or MPOD or stim trackers or any of those devices. Whereas and also you can use LSL for sharing with other hardwares. But today what I'm going to demonstrate is how to communicate, establish a communication between SuperLab and BioPack AcqKnowledge. So hit this button, there'll be a window appearing before you. What you do is you just click it on, it'll take a few seconds and the connection is established. In case you are connecting with another computer in which SuperLab is running, you need to use the LAN connection. So it takes little more time. You need to put the IP address. So that's how the connection is established between SuperLab and AcqKnowledge. So now at this point of time, whatever would SuperLab present, it will be sending event markers to AcqKnowledge. And if I have to show you, these event markers will appear in this bar. This is known as global bar, where all the events appear when you run the experiments. So let's run the Stroop task. So all the connections are intact. Now what I'll do is I'll start running the AcqKnowledge software so that it records. And then what I'll do is I'll run one of the blocks here. So, and then you can also save the data. So you can, the next option would be, it will tell you exactly which folder you want to save all the numerical data which will come from SuperLab. So you save it, and now the paradigm would start. So it will give you options, like as Hisham was talking, you can for each color you can, like in case of keyboard, you can give specific keys to specific colors. So the subject has to press those keys when the ink of the color appears the way it is, like for example red Z, green X, blue C, and purple V. Now let's start the experiment. So what I'll be doing is, in this setup, I'll run the Stroop task. Whenever the word appears, I have to pick up the color, the ink of the word, and press the button as fast as possible. And I'll be in the background, AcqKnowledge is recording all my biological parameters. So Hisham has created maybe around 20 stimulus presentations. I'll quickly run this, and then I'll show you that how all these events were picked up. So it is the end of the trial. So let me show you the AcqKnowledge here. So this will be, it will squeeze everything into one screen. So here at this point, I started the experiment here, and around this point, I start the experiment. Before that, it was recording a baseline. Now, what I'll do is I'll show you the interpretation part. So I need to magnify small window here. So if you see here, there are a lot of markers which are coming to the global bar here. So let's see, I'll magnify this one here. So now these markers, if you see, they are all downward arrows. So if you want to see exactly what was the temporal sequence of appearance of these markers, event markers, you can go to drop down menu here and select event palette. So it will show you all the markers here. So let me again minimize. So select a bar here, start from here, and drop down menu again, event palette. So here, what Hisham also showed, that the event list is very detailed. When was the experiment done? So all events will come to the AcqKnowledge, and they'll be listed in this event palette. So this will be very helpful depending on your research design. So you can pick up all the temporal sequences, when the cue was given, when the word was presented, when subject responded. So this is the event palette Hisham was talking about. Now, next step is, let me magnify a little bit here, so that you understand what's happening. So, okay. So I magnified too much. Let me magnify a little more. Yes. So if you see here, this channel is showing button press. So whenever the stimulus appeared, there was a button press here. Now, if you point toward these event markers, this was the onset of the text. And here the subject pressed the button. So this will give you approximate reaction time. But when you press the keyboard, SuperLab will also generate a file which will be saved in one of the folders in the beginning I mentioned. So you know exactly how much was the reaction time. Now these event markers can also be used for doing various analysis. So you can create focus areas, or you can also do averaging. But because all these global event markers are of similar nature, symbol is almost similar, downward arrows, you can edit them. Let's take this event and edit. So you right click this event. So there will be this function that is edit events. So now we are exactly on this event marker, and here are the list of options what you can do with this event. So we need to change the symbol. So let's make it a star. You see now it's a star. So like this, suppose later on you want to create focus areas between only stars, or you want to average around these stars. So editing these events will be very helpful. Now, what I'll do is I have recorded another file. So here is the file. So the first part is the baseline. And then what I did, I took deep inspirations and expirations. And then I studied the effect on what you call EDA response here, right? Now, this is when the inspiration started. These markers are the inspiration, beginning of inspiration. So I used hand switch to show it in one of the channels. So this is during inspiration. There was a change in the EDA response. And also you can also study the pulse and also ECG and heart rate variability when inspiration took. So this will give you what you call sinus, respiratory sinus arrhythmia, because that's what happens when you take deep inspiration. There is change in heart rate and also sympathetic output. Now, if you go further, this is the Stroop task when the Stroop was presented. This from here to here. So whenever the target stimulus appeared, I pressed the hand switch. And also, if you see here, in the global panel, you can see the events are also there. So these are the events. So the next trial. So you can use these event markers as per your research and study design. And then if you see, next was I presented a startle stimuli in this focus area from here to here. And what I've done is I have looped the auditory stimuli into the AMI 100D. So whenever auditory stimulus comes, startle stimulus, so there is also a response in one of the channels. And then at the same time, the global panel is also giving a stimulus input. So you have many ways you can find out exactly when the stimulus was presented by the computer, by the software, and when it was perceived by the subject. So this will be very useful, especially during the analysis of doing these stimulus response studies. Now what I did is, although this is beyond the scope of this webinar, I did a little bit of analysis of these records which I uh, uh, recorded. So the first one, if you see, this is the focus area, and I'll just share with you how to create focus areas quickly. You select the area you're interested in, right click, and then go to this menu, that's create a focus area. This is how I created these focus areas. So the second one is during inspiration. So what was happening during inspiration? there was, clear-cut EDA response. Let me hide the other channels so that you can see the EDA responses. So whenever I was taking inspiration, so you see there are these water drops. So they are the peak of the EDA response, and there is beginning and end. So whenever there was inspiration, there were these responses. And then in the next one, if you see, whenever Stroop Task, the target stimulus appeared, that is, in this scenario, it was naming the color of the word. So there was also some phasic responses seen here. What I've done is I have edited the events. I made them stars so that later on, if you are interested in doing some analysis, you can use these as event markers as a reference if you want to create focus areas, or if you want to create, if you want to do averaging. So if you see here, this is the point of time when the target text appears. And here, if you see, this is the peak of EDA response. In a similar fashion, you can analyze ECG. You can also record, like, you can record EMG, EEG signals, and then also study later on what will happen to a stimulus and response. So this is how we are able to, communicate with the SuperLab. And it's very easy with now NDT, or with working in the same computer, that is the SuperLab and AcqKnowledge. So there is no need of a second laptop, and also there's no need of many other gadgets which otherwise are needed, like response pads, or C mode, or M pod, or C pod. So the requirement, of course, will be there. If you have a design where you need to play in two different computers, you can still use TTL signals, which is of advantage. The next paradigm which Hisham created, he created this one. First, it's an oddball paradigm, where targets and non-targets are given. Subject has to press the button when the target appears. So these are the sounds. When the pitch sound comes, the subject has to respond by clicking the head switch. Now, this paradigm basically helps in understanding selective attention. So what I'm going to do is I'm going to use the paradigm which was created by Hisham here. So here is the oddball paradigm. So every time you switch on the SuperLab, you want to communicate with AcqKnowledge, you have to always go to experiment and make sure that BioPack AcqKnowledge is ticked in. So let's see whether it's still there. So you see here, because I opened another paradigm, so we have to again establish the connection between SuperLab and AcqKnowledge. So let's test the connection again. So it's now successful. It hardly takes seconds. So it's done. Now what I'll do is I'll open AcqKnowledge on the other side. So I have created a paradigm here, and also picking up from various AcqKnowledge channels. So let me go to the setting first, because in this scenario we want to pick up EEG, because we want to do averaging later on. So here, if you see channel 6, it will pick up EEG. It's already connected to the AMI 100D, but I'm not wired because it would have been impossible while giving the talk. So now channel number 6 is EEG. I'm not removing other channels. So what you do is simply run the experiment again. Go to the play mode, save the collected data with specific file, run the selected blocks, and start the. So before that, you also need to run the AcqKnowledge, so that everything is recorded. So it has started recording. Now let's run the SuperLab experiment. Okay, so these will be two sounds, targets and non-targets. So this is the end of the trial. This is not a complete trial. This is just an example, as Hisham was telling you guys. So I stop it here. Then in the background, the AcqKnowledge is running, recording all the relevant channels. So you see here, whenever I was pressing the button, there's a response of the hand switch here. And if you check here, so this is the target, this is the non-target, this one is target, non-target, right? So this one is the target which appeared, to which I responded. So let me enlarge a little bit so that it's clear to you guys. So here, this one and this one is the target stimuli. So in the global panel, you'll find all event markers coming. Now in this scenario, especially in this case, we are going to average the EEG response. So there is in the end, in the last channel is the EEG, which is noise because I'm not connected. But then what we need to do is we need to edit, because both target and non-target have similar event marker symbols. So as we did last time, to make all of them, all the targets as stars like this. So this will help in analyzing. To just save time, I have already a pre-recorded sample tracing with me. So here on the top, if you see, there is a global bar where you find targets and non-targets coming one after the other. And the targets are stars here. If you see here, what I've done is I've edited them and all the targets are shown here as stars. And then you have EEG signal, you see, in all these 5 channels picked up from FZ, CZ, PZ, and C1 and C2. So, of course, we also picked up EOG. If you see here, we have picked up EOG. And in the beginning, when the recording just started, there was a lot of interference or noise from EOG. So by looking at the AcqKnowledge tracings, you can see exactly whether there is any noise coming from EOG, and later on you can filter it. Now what I'm going to do is I hope you understood that all the target stimuli, response to target stimuli, I'm going to average. In the response global panel, what I've done is all the targets are marked here as stars. Now what I'm going to do is average. So this data is already filtered. Otherwise, you can transform the data by using various digital filters. There are specific recommendations for each signal. And then you go to analysis. There is an option of ensemble average. Pick it up. So now what we are going to do is we're going to average maybe one of these channels. Let's say FZ. Now we are going to average based on the events in the global panels. So we'll pick up global panel events. And what is the event? Event is a star. Star is when the target is appearing. If you want to average, uh, let's say non-targets, you can choose flags, because that's when the non-targets are appearing. But in this scenario, we'll select stars. Where are these stars present? They're present in the global event only. But they can also be, these event markers can also be present in the channels. So you can select whatever is your study design. So I'm choosing here global event only. And then what I'm going to do is I'm going to average the whole of the graph. So I hit OK. Now, let's say, because this response is vertex maximum, so let's choose CZ. And now it's asking me, we're going to average around start, all the EEG, which is around the start. So it's asking me, we have to select the time duration before and after the start, which I'm going to do now. So I'll pick up 100 milliseconds before and 500 milliseconds after the start. I hit the button average. So it's averaging quickly. So one of the channel is averaged. Just for better viewing, I'll just smoothen it so that you're able to see the waves. So this is how the SuperLab is presenting the stimuli. AcqKnowledge is picking up various biological signals. In this scenario, we picked up EEG. Could have also picked up maybe eye tracking, or you could have picked up functional near infrared cerebral blood flow changes, or maybe EMG. And then in this case, especially, based on the events which are coming directly from SuperLab, we have averaged the EEG, and what we have found is typical N1, P2, N2, P3 response. So this is telling about that it takes around, in this case, maybe around 410 milliseconds to decide whether this was a target or not a target. This is known as typical event-related potential, which is known as P300. So this is how we are going to use various biological signals in response to the stimulus input which is coming from SuperLab, and then do analysis. Now, if I show you in the analysis window of AcqKnowledge, there's so many functions possible. I just demonstrated only one. So depending on your research paradigms, your study design, you can pick any of these and do your study. So this was in brief about how to use AcqKnowledge and do the analysis part. Thank you very much. Brenda?
All right, wonderful. Thank you, Sanjay. I am going to switch screens to me.
Yep.
And I just want to share a couple of things before we close. Fraser's been doing a great job answering questions in the background, so I'm not going to ask you guys any additional questions. But I did want to let you know on our website, you can just search for SuperLab up here and get to SuperLab. You can also search for network data transfer and get to the BioPack Developer Acknowledge License Pack, which is how you would access the network data transfer. By the way, network data transfer works with any of the third-party products to connect with AcqKnowledge, including SuperLab. So it's a great opportunity to use with other products as well, such as Research Ring. And finally, I just want to share that you can contact your local sales representative who can help you think through your different needs. You can reach us through the contact us form, or if you want to know who you're interacting with, you can scroll down on any page of our website, click this button. My local sales rep is Amy Walker because I'm in California, but yours will be displayed here on this page. And if you fill out this form, it will automatically send it will automatically send an email to that person, and they'll get back to you as soon as they can. Okay, now I just want to say that we have over 100 webinars on our website. Our next one is November 21st. The email is going out about that today, so feel free to join us in a couple of weeks. And we host webinars almost every month of the year, so check back on our website, biopack.com/webinars, to watch any past webinars or sign up for future webinars. We also host workshops in regions all over the world. Our next workshop is December 12th in Copenhagen. You can use that QR code if you're there locally. You can sign up for that. We also have some workshops coming up in Germany. We just finished some workshops in China. So if you're interested in hosting a workshop in your area and inviting some of your regional colleagues, reach out to us and let us know. We'd be happy to come to you and spend the whole day with you, training you on the equipment and sharing our tips and tricks with you as well. All right, today's webinar was recorded, and thank you, Sanjay and Hisham. That was a great
Thank you,
presentation.
Brenda.
Yeah, I know it takes a lot of work to put these together, so thank you for all the time that you put into that. We'll email everyone a link of today's recording and the slides. When you close the webinar window, you'll see a survey. Please complete that so we have your feedback and ideas for future webinars. And you may visit biopack.com for additional references, including screencasts on specific features and analysis tools, application notes with step-by-step instructions, and of course, information on upcoming events on our calendar. It looks like, oh, Daniela, the recording is usually available in the next day or two, and we'll send out that email. You can also access the recordings on our website, biopack.com/webinars, and you can access any of the recordings there. Anything else before we conclude, Hisham and Sanjay?
Just a quick note about the recording. When you run an experiment in SuperLab, it automatically starts recording on AcqKnowledge. You don't need to separately go and record that. You don't need that separate step, basically, right? And if AcqKnowledge is not detected, SuperLab will tell you, hey, I can't find AcqKnowledge. So basically, it makes sure you're not wasting time, you're not wasting data, just smooth operation.
Okay,
Yeah.
great.
That's a very good feature. And Brenda, I like to say to audience that today's webinar had different focus. It was about SuperLab and AcqKnowledge communication, and it was not about how biological signals are analyzed. So I may have skipped some critical steps. So those who are interested can watch our webinars. There's in-depth, procedure given in those webinars. Thank you, Brenda.
Okay, no problem. I will go ahead and show you the webinars page. So here, biopack.com/webinars. You can scroll down and find additional webinars on StimPres and SuperLab here, where we go into more detail about, and AcqKnowledge as well, where we go into more detail about analysis. Okay, well, thank you both again. Thank you to the audience for attending and for asking some great questions. Thanks to Fraser for answering most of those questions. This concludes today's webinar. Have a great rest of your day, everyone. Thanks and
Thank
goodbye.
you. Bye-bye.
Bye.